In this video, we'll begin the discussion involving animal virus replication. Animal virus replication is a five-step process, typically. It is broken up into the 5 steps of attachment, also known as adsorption, penetration and encoding, followed by synthesis, followed by assembly and maturation, and then finally release. Like I said, in this video, we'll begin that discussion, and what we're going to focus on is the attachment step. The attachment step in the animal virus replication cycle actually involves the virus binding to receptors, typically glycoproteins, which are some combination of a protein as well as a sugar on the surface of that particular cell. In many cases, what you'll see is that these animal viruses have to bind to multiple receptor attachment sites. In other words, their spikes typically have to attach to multiple glycoproteins. Ultimately, what this means is that, these animal viruses have a high degree of specificity. Now, I don't usually like to say specificity because it's a term that usually comes up later on in the immunity discussion, but ultimately the consequences of this multiple glycoprotein receptor attachment situation is that the animal viruses will not be able to infect multiple cell types in most cases, nor will they be able to infect multiple animals. As you can see down here, for instance, most viruses, like the measles, which can infect humans, the same virus would not go and infect a dog. Reason being is because the receptors, the glycoproteins on the cell surface of a dog, are not going to be the same as that which would be found on a human. Now, there are some viruses that do negate that trend, and one prominent example would be the rabies virus. It can be transmitted from a dog to a human, but by and large, because of the fact that these receptors that are bound are unique from species to species, what you're going to see for the most part is that most viruses infect one species and one species only. Not only that, but because the receptors are so specific, you'll typically see that they also have specificity in regards to what type of cell they will infect. For instance, in humans, the influenza virus will preferentially affect cells within the lungs and not infect cells within the liver or bone, for instance. It's all because of the receptors, and therefore you will have specificity. So, to summarize, because I'm going to make a video for each one of these steps, you do have those 5 steps that I mentioned to you earlier. The first step being attachment or adsorption. When you hear the term adsorption, think add to. That's basically that's what reminds me, or at least triggers me, to think of attachment. Keep in mind that these animal viruses attach to specific glycoproteins within the cell membrane of these animal cells. And because of the need to attach to multiple glycoproteins, it gives these viruses a high degree of specialization or specificity. In other words, they're not going to target multiple cell types in multiple species in most cases. Like I said, there are some exceptions to that rule, but by and large, that is the rule.